So today is quite special. We have our special guest from China, Professor Xiaomei Yan. She's a distinguished professor of Xiamen University in China. Xiaomei Yan is an analytical chemist, and her research lab has developed nano-flow cytometry that can detect single EVs, viruses, and silica beads as small as 40 nanometer, 27 nanometer, and 24 nanometer. She's also a co-founder of Nano-FCM. So today her talk is about EV labeling, method and cautions learned from flow cytometric analysis of single EVs down to 40 nanometer. Please welcome Xiaomei.
And thank Carolina for the nice introduction. Hello, everyone. Like what Carolina just said, yes, we have developed probably the most sensitive nano-flow cytometer. This instrument provides us a very unique opportunity to have a good view of the have a good view of the full picture of EVs. And today I would like to share with you the perspectives. I add isolation besides the labeling. So today's presentation will cover these 6 topics. First, I would like to talk about why it is important to use flow cytometry for single EV analysis and how difficult it is. I don't know why it pops up every time. So we all know the EVs are nanoscale membrane-bound vesicles. How can if there is someone comes in, then this button will always shows up?
Yeah, but don't worry, we don't see it, so it's okay.
Oh, you don't see it? Okay, that's
we
fine.
don't see it. Yeah.
Okay, good. Okay. So with most of the EVs smaller than 200 nanometer in size, but also, even though it is small, but it has a very large chemical complexity about the cargo molecules, and the EVs are also highly heterogeneous in size, cell origin, and the chemical compositions. And also the physiological environment in which the EVs present, such as plasma or the cell culture medium, is quite complicated. For example, in the plasma, the particle concentration could be 10 to the 12 to 10 to the 13 particles per milliliter. And for EVs, the concentration is only 10 to the 8 to 10 to the 9. So detecting single EVs in plasma is like finding a needle in a haystack. So, and also there is a considerable overlap between the EVs and the lipoproteins in both the size and the density. So normally people use ultracentrifugation to isolate the EVs for the downstream analysis. So normally cryo-TEM or TEM and NTA and tunable resistive pulsing techniques are used for single EV analysis, but none of them can reveal the biochemical properties of EVs. And also for NTA and the nanopore techniques, they can hardly detect EVs smaller than 70 nanometer. We know that flow cytometry is a key technology for the multi-parameter analysis of single cells. It has many advantages, but the applicability of flow cytometry to the nanoscale EV analysis presents many challenges. One is the limitation of the instrumentation, and also the lack of robust methods, and also how to interpret the data. Based on a recent report, for light scatter based EV measurement by flow cytometry, only couple highly sensitive flow cytometer enables the detection of EVs of 300 to 600 nanometer in size. So we really need to develop highly sensitive flow cytometers to decipher the heterogeneity of EVs. And earlier this year, a group working on EV flow cytometry, researchers from 3 society, ISEV, ISAC, and ISTH, published a position article in JEV to report a consensus framework for standardized reporting of extracellular vesicle flow cytometry experiments. I do recommend, if you are interested in using flow cytometry for EV analysis, please refer to this article for standardized reporting. Okay, as I just mentioned that there are only several highly sensitive flow cytometers that can detect EVs of 300 to 600 size range. But what about the true size of EVs? So we use the cryo-TEM, the most accurate method. We measured the size, EV size distribution for EVs isolated from human colorectal cancer cell line, HCT15, from the cell culture medium, and also from a urine sample. We can see from this size distribution histogram that the population of EVs larger than 300 nanometer in size only takes a very small fraction. For example, probably 8% or even less than 22%. But why can still flow cytometer measure it? Let's assume the total EV concentration is 1 × 10⁹ per milliliter. If the EV is larger than 300 nanometer, takes only 1% of the total population. So the EV is larger than 300 nanometer, the concentration would be 1 × 10⁷ per milliliter. This concentration is good enough for flow cytometry analysis, as normally for cell analysis, the concentration we used is 1 × 10⁶ to 10⁷. Even though flow cytometry can detect EVs larger than 300 nanometer, no problem, but we need to be aware that conventional flow cytometry does miss the majority population of EVs. So this is the case. So I fully agree with what Dr. Philip said. The actual cellular vesicle analysis by flow cytometry is like you can only see the iceberg. So we really need some submarine that can dive down to the sea to be able to see the full picture of the EV. Fortunately, but I also want to highlight, actually this is a very challenging task because based on the Rayleigh scattering theory, the scattered light intensity of single particles scales off with the 6th power dependence of particle size, which means with the particle size decrease, the intensity will soon drop below the background. We also did a theoretical calculation. The light producing power of a single 25 nanometer silicon nanoparticles actually is even smaller than a single alloxofluor 532 fluorescent molecule. On the other hand, besides the particle size effect, the refractive index also significantly affect the scattered light intensity of EVs. We can see here the different material polystyrene particles, silicon nanoparticles, and EVs, they have different refractive index. For example, for a 60 nanometer polystyrene nanoparticle, its scattered light intensity is 4-fold brighter than the same size silicon nanoparticles. And the silicon nanoparticle, the brightness is another 3.5-fold more brighter than EVs. For example, here, if we look at the window, the side scatter brightness of 160 nanometer polystyrene beads actually fall between the intensity between 300 and 400 nanometer silicon nanoparticles. So we know detecting the small size EVs, let's say down to 100 nanometer or 40 nanometer will be very, very challenging. Fortunately, we have developed this technique, this nano-flow cytometer, that enables us to detect single EVs as small as 40 nanometer. So we at Xiamen University, during the past 14 years, we have been working on the development of nano-flow cytometer by integrating radius scattering with the strategies for single molecule fluorescence detection in a schist flow through 3 stages of technology enhancement. In 2014, we reported the detection of single silicon nanoparticles of 25 nanometer. And also if it is for gold nanoparticle, because of the plasma resonance effect, we can detect 7 nanometer gold. And the detection limit for fluorescence is about 3 electron molecules. So in the case of EV analysis, we reported protein profiling and sizing of single EVs as small as 40 nanometer, and the analysis rate was 10,000, up to 10,000 particles per minute. And the resolution and accuracy for EV sizing was comparable to that of cryo-TEM, but is more statistically representative. And the protein expression can be analyzed with the sensitivity of single Vesicles molecules. So this instrument enables the simultaneous detection of the side scattering and the 2 fluorescence signals of single EVs. Because even with the ultracentrifugation for the EV isolates, there are also some lipoproteins co-isolated with EVs. So we would like to know the purity of EVs. What we used is we used Triton X100 to lyse the vesicles. If it's 100% vesicle, after the Triton treatment, you wouldn't be able to detect particles. So by enumerating the particles before and after Triton treatment, we can quantify the purity of the EV isolates. We can see that for 3 samples of EVs isolated from cell culture supernatant, by using ultracentrifugation, the purity was around 90%. But for the EVs isolated from platelet-free plasma, the purity was 70%. This is easy to understand because of the there are a lot of lipoproteins coexist in the isolation from plasma. So this slide illustrates how we measure the EV size. So first, we synthesized size reference standards by using silicon nanoparticles to construct the calibration curve that can convert the scattered light intensity of single particles to the diameter. I also want to emphasize that due to the refractive index difference of silicon nanoparticles and the EVs, we used the mean theory calculation to derive the correction factor for the fluorescence brightness. So here we can see this is an EV sample. We can get the side scattering burst area distribution. This is only the intensity. And then we use the mixture of the silicon nanoparticles to construct this calibration curve that can converts the scatter light intensity to the particle size. This is also refractive index converted. So with this calibration curve, we can convert the brightness of each individual EV particle to the size. So here, you can see this is a particle size distribution for the EVs. Meanwhile, for the same sample, we did the cryo-TM measurement. We took 80 TM graphs and counted more than 1,000 particles to get this distribution. It took us more than 2 days. We can see the distribution profile obtained by Nano-FCAM faithfully resemble that of the cryo-TM. Okay, let's do a resolution comparison among 3 techniques, the TM, Nano-FCAM, and NTA. So first, for the silicon nanoparticles, the Nano-FCAM can completely resolve the 5 populations of different sizes of silicon nanoparticles, whereas the NTA cannot resolve them. For the EV samples, the peak position and also the shape obtained by Nano-FCAM and the cryo-TM, they are very comparable. But for the result obtained by NTA, you can see both the shape and the peak position deviate largely from their corresponding values to those obtained by cryo-TM or Nano-FCAM. So the good accuracy and the fast speed and the statistical robustness, they are the key advantages of using flow cytometry for EV size measurement. People are very interested in analyzing the surface proteins of EVs to identify their cell origin. So we analyzed the CD9, CD63, and the CD81, the 3 most popular markers for EVs. We can see from the dot plot that for this sample, the purity is around 90% of the EVs, but we can see those CD marker positive EVs, they're only around 50%, which means not every EVs express these markers. Why can I say it? Because our instrument is so sensitive, it can detect a single antibody that conjugated by phycoresterine. So this signal is a single antibody. So if we calibrate the brightness of this CD fluorescence with the fluorescence of the single antibodies, we can quantify the copy number of how many CD markers expressed on each EV molecule, each EV particle. So here we got for the CD9, so the median value, only 5 copies. Some are for the CD63, 6 copies, and it can vary from 2 to 15. So we can make a quantification of the protein expression on single EVs. So this instrument is very sensitive. So what can we learn for the EV isolation? So we compared, we used the Nano-flow cytometer to compare quantify the quality and the efficiency of 6 extracellular vesicle isolation methods, 5 commercial kits, and compare the performance with the classical ultracentrifugation. So by measuring the particle concentration, size distribution, the purity, and also the surface proteins. So these kits are listed here, and the isolation of principles include for ultracentrifugation, sedimentation, and polymer for the kits, polymer precipitation, size exclusion, membrane filtration, and membrane affinity. So first, let's look at the size distribution of platelet-free plasma. this is just the plasma prior to any isolation. And here are the size distribution profile of this PFP sample used as the starting material, and use different kits or method to isolate it. And this is a TEM, the representative TEM image. We can see for the PFP sample, so the size of the particle is a very narrow distribution, and the median size is 56 nanometer. But for the EV's preparations isolated from PFP by ultracentrifugation, we can see the particles covers a large size range from 40 nanometer to 200 nanometer, and the median value was around 81 nanometer. But for the EV preparations isolated from other kits, you can see those shape or distribution, more or less, they closely resemble to the PFP. We also measure the particle concentration from these isolates. Pay attention, this scale is in log scale. And if we look at this table, particularly, we can see that, so in PFP, the concentration is quite high. It's like 1 × 10⁻13 per milliliter. For the EV isolates from ultracentrifugation, so which means EVs only takes a very small fraction of the particles, of the total particles in PFP. And the particle concentration for the EV isolates from these commercial kits, they are about 2 to 4 orders of magnitude higher than what isolated by ultracentrifugation, than what these particles are. So what we did is we depleted the vesicle from PFP by ultracentrifugation at 100,000 G, 18 hours to deplete the EVs, and then use this vesicle depleted PFP as the starting material, and also use ultracentrifugation and those 5 commercial kits to isolate. Very interesting and very ironic. Presumably, there should be no EVs inside this vesicle depleted PFP, but those commercial kits also use very, very high concentration of particles, and the particle concentration almost the same as when you use PFP as the starting material. So we also measure the purity. So if the particles isolated from the plasma preparation vesicles EVs, if we use Trita X100 to treat it, there should be, you would see a significant decrease of particle numbers. So that is true for ultracentrifugation. And the purity, what we measured as shown here, we can see for the ultracentrifugation, the purity was around 78%, but for the other commercial kits, it's quite low. For the Exo Easy, we cannot use this approach because Triton treatment will let some large aggregate break apart. That means it's not the particle counts not decrease but increase. So this method is not applicable to the purity measurement of EVs isolated from ExoEasy. And normally we say in the published paper or the manufactured data sheet that showing the performance of the kits is that, okay, their isolates has very high concentration of those CD markers. However, what we met, which means they have a high recovery of EVs, but this is what we did. We put the starting material of either plasma or vesicle depleted plasma, and we use those kits and ultracentrifugation to isolate it, and we run the Western blot. We can see for those CD markers, the pattern of the vesicle depleted plasma almost comparable to what we got from the PFP. Presumably there should be no EVs inside, but we can still see the high concentration of those CD markers, which means those markers not only reside on the surface of EVs, but are also present in presumably some other non-vascular materials. So we really cannot use merely just the Western blot analysis result to say, okay, the kid has a high recovery of EVs, because this is ensemble average method. It cannot differentiate whether those proteins are associated with the vesicles or associated with other non-vascular materials, or simply just reside in the solution. Okay, this is very interesting because we compared those surface markers for EVs isolated from cell culture supernatant and from the plasma. So besides those three classical markers and PAs, we also added those CD markers. Those are representative of those cell type for EVs secreted by this. So you can see for the EVs isolated from cell culture supernatant, EVs expressing those cell type specific markers are quite low. But on the other hand, for the EVs isolated from plasma, you can see particularly for the CD235A, this is a marker for erythrocyte. So the percentage was around 60%. This can be attributed to the large, the abundant presence of erythrocytes in the blood. So the notes for EV isolation, there is a high degree of non-vascular contaminants exists in plasma isolation by kits. And compared to the commercial kits, single ultracentrifugation demonstrates great efficiency in separating large quantities of non-EV contaminants. And we can also use a combination of ultracentrifugation followed by gradient density ultracentrifugation if you want better or higher purity EVs. Okay, what about the EV labeling? Because there are no generic protein markers for EVs, so a more general approach is to use a lipophilic fluorophore to incorporate them in the EV membrane to label the EV for either tracking using fluorescent microscopy or EV detection by flow cytometry, because this double-layered lipid membrane is the most common feature of all EVs, and the PKH or DRI series dyes are the most common dye used. So we use the PKH67 as an example to illustrate the EV membrane labeling. It is recommended to add the EV diluent to the PKH diluent of equal volume to get a homogeneous staining. Normally, we use 1 to 5 micromolar PKH67 to stain this many concentrations of EVs, and the EVs can be well detected above the background. We highly recommend to remove the unbound EVs. People may ask that washing away the unbound dye may take times. If we use a tabletop ultracentrifuge, that can shorten the centrifugation time from the 2 hours to 70 minutes. But cautions need to be aware when we do EV membrane labeling. The first is can all the EVs be labeled? What is the effectiveness of labeling? And also those dye molecules could aggregate and form micelles that resemble the structure of EVs, and also whether the contaminated lipoproteins can also be labeled. From this dot plot, let's say for EV, the purity was around 88%, but at the saturating concentration of pKh 6.7, the labeling ratio was only 67%, which means not all the EVs can be fluorescently labeled with lipophilic fluorophores, even under saturating labeling conditions. Okay, and also we know that those lipophilic dyes can form aggregates that resemble the structure and size of EVs, but how severe is this effect? Okay, so we found out upon washing away the unbound dye, there was an 8-fold enhancement in the fluorescent particles compared to the reagent controls. Actually, the particle number arised from the aggregate dye molecules is much smaller than this number in the reagent control, because a lot of dyes would bond with EVs. So if the unbound dye are washed away, we normally don't need to worry about the aggregation effect. Also, due to the considerable size and density overlap of EVs with lipoproteins, we tested whether the residual lipoprotein would also be labeled by those membrane dyes. This is a VLDL, a very low density lipoprotein. We can see that almost every particles can be labeled. So for sure, for the EVs isolated from the plasma, there, if you want to use membrane labeling, there are also some labeled lipoproteins. And also, people are interested in using the cyto-RNA select to stain the EV for the EV-RNA tracking. This is the only commercially available RNA dye. Even though the fluorimetry experiment demonstrates that it has much higher specificity to RNA compared to DNA, but our experiment indicates that this dye can also stain single DNA fragment very bright. This is only, each peak represents a single 2,000 base pair DNA fragment passing through the laser beam. And also this experiment, if we use this dye to stain the EVs and use DNase 1 to treat it, which only digests double-strand DNA, we can say after the treatment, the events of fluorescence even dropped significantly. So this confirms again that the cyto-RNA select also stain DNA. So this can now be used for EV-RNA tracking. Also for the surface protein labeling, we use we also need to be aware that if one is we need to, before pipetting the antibody, we need to do a pre-centrifugation to avoid the potential aggregation of antibodies. And also it's highly recommended to remove the unbound antibody. It can be done by either ultracentrifugation. And also we need to be aware about the stereohindrance effect when we select the ligand and the fluorophores. Okay, normally we use this quantity of antibody to stain this many EVs. So you can see there could be a lot of free antibodies reside in the solution. If we don't remove them, they could cause significant fluorescence background that would easily mask the detection of small EVs on which the proteins are only medium abundant proteins or very low abundant protein will be very difficult to detect. And also we need to be aware that because some people are interested in multi-protein labeling for a single EV. So for a 10 micron size compared to a 10 micron size cell, 100 nanometer EV, I mean, the surface area and the volume was this much reduction. And also, the antibody is big, is about 50 nanometer, and fecal reservoir is also very large. So spherical hindrance effect will definitely block the efficient labeling of multiple proteins on a single EV. Probably we need to consider using some small size ligands such as FAB domain or aptamer, or replace fecal reservoir with the organic dye. Okay, this is the notes. I will not repeat it. We also applied the single EV analysis for the cancer diagnosis. So the CD147 is a putative marker for EVs secreted by colorectal cancer cell lines, and we confirmed this by using nano-FCM and a Western blot. I really want to highlight that you will need to use a lot of quantity of cell in order to extract EVs for the Western blot analysis. But we only need 1/50th amount you need for Western blot analysis. That is enough for a nano-FCM experiment. Okay, first of all, we measure the particle concentration in the plasma. There are 32 healthy donors and 37 colorectal cancer patients. We don't see significant difference between these two populations. For individual people, the range of concentration can vary 40 to 50 fold. But later, if we detect the CD147 specific EVs detect the concentration, we found a significant difference. It's about 7-fold difference between the healthy donors and the colorectal cancer patients. And if we took a more clear view about the cancer patients around different stages, we can see even at the first stage, significant differentiation can be observed from the healthy donor. And for 16 patients going through the surgery, we found that 13 among 16 the people going through surgery, we did the EV measurement 1 week after the surgery. So 13 among 16, we saw a significant reduction of the CD147 positive EV ratios. So by providing rapid and multi-parameter analysis of single EVs down to 40 nanometer, nano-flow cytometry can determine the specific subpopulation by measuring the protein expression. It can be used for disease diagnosis and treatment. Monitoring can also help to standardize and optimize the isolation protocol and for the quality control of EV products. Besides applying the nano-flow cytometry to single EV analysis, we also applied this technology to bacterial, viruses, nanomedicines, and mitochondrial. And here are our publications. So this technology is really powerful. So enable the researchers in EV field or some other nanoscale particle analysis research field. We have made great efforts to bring this technology into commercialization. So the Nano-FCM company was, this startup company was founded in 2014. And the first instrument was brought into the market in 2014. And the first unit was sold to Kodak Biosciences. So this is a part of the custom list. Most of them are using the nano-flow cytometer for the single EV analysis. And this is more information about the company. We also have a subsidiary company in Europe and the USA, and you are very welcome to contact them for more information. In the end, I would like to thank the funding support from the NSFC and thank our group member for the hard work. And also thank you very much for your kind attention. I will be very happy to take your questions. And also welcome to Xiamen University. And this is the most beautiful campus in China. The Xiamen University is also the top 20 universities in China. Okay.
Thank you very much. I found the key to unmute myself. I was not unable. Thank you so much for a wonderful presentation. You've really done a great job. It from the first publication, it's taken 6 years to now to really get it out.
Back in 2014, we demonstrated it can detect single nanoparticles of 25 nanometer. And in 2016, single viruses about 27 nanometer, and 2018, single EV 40 nanometer. Yeah.
So when was the first time you had your first provisional machine?
Your
first prototype?
Prototype, as a prototype was in September of 2014, actually.
Oh, really? Okay.
Yeah. But before we brought this into the market, we need to make sure it's stable and with good performance. So a multiple generation of prototypes, finally. Yeah. And thank you, Yang, for the great trust. And you are my first customer.
Yeah, I bought 2 machines, right?
Yes.
So for different organizations. So the first question is from Rosella. Crescitelli, please unmute yourself, or and you can ask your question, Rosella.
Okay.
Yeah, I work in Yan lab, and we have the machine, as you said.
Okay.
Yes, we are trying to use it, and we are struggling with it. I mean, it's great, but it's not super easy to start to work with it because we need to optimize a lot of things, and we feel that we need to be really well. We have to know really well how the machine works to optimize the stuff, to align it. So I feel that is not very friendly. Anyway, my question is about EV sites, because it was not clear to me which subpopulation you are working with, which EV subpopulation. Because using the machine, I know that before to start the sites analysis, we have to use a specific kind of beads with different sites. So basically, we have to know if our samples is enriched by large vesicles, microvesicles, or small vesicles, exosomes. So from your presentation, it was unclear to me which kind of population you use. I suppose small vesicles, exosomes.
Oh, okay. Sorry.
Okay. No, sorry. And just if you can clarify that, because it was a big surprise for me, because our point is that when we use really new sample, we really don't know if our sample is enriched by large or small vesicles. And this is what we ask to the machine. Please let me know if I have large or small vesicles enriched in this kind of sample. But in this case, as for a lot of different machines available, we have to, in a sense, we have to know if we have large or small vesicles in our sample. Can you comment on it? You are working on it, or?
Oh, sure. It depends on how you probably isolate it. So what we use ultracentrifugation. So most of the, so we first use the 800G, 5 minutes to get rid of some large cell, and also 2000G, 10 minutes. And then we use 100,000G, 2 hours to isolate the EVs. And when we put those isolated EVs on the instrument, we normally see the size is smaller than 200 nanometer. But in our paper, let me see, it's the JEV, the new paper, the comparison of the kit preparation. So in the supporting information, we have data shows if you have large size particles or large size EVs that saturate the detector, you would still be able to see it. It's just saturating, you wouldn't miss this population. But if you don't see the pipe up on the very large size saturating, which means there are not large particles in your sample. Did that answer?
Okay, and yeah, sure. But have you ever tried to mix large and small EV and see if the machine is really able to show two really clear different peaks for large and small?
Okay, I don't know, you mean large or small is which scale for the instrument?
Let's say exosome and microvesicles. Maybe it's easier.
Nobody can differentiate. Nobody can differentiate.
Yeah, I know, but we know that we have different sides. So this is my question. If the machine is able to really look at two or more peaks in a complex sample.
Okay, we use, if we put silicon nanoparticles, which has very narrow distribution, right? We can
I know.
Them and analyze very good separation. So it's the same principle. If the only thing is the large size particles would saturate because it's also the exponential dependence. A slight increase in size would enhance. But in our system, if it saturates, if you really want to know if there is large size particle, what you can do, you can tune down the laser power.
Okay.
Your option, and also you have some density filter, so you can do this. So larger one is not a problem. Normally, I think smaller one, because those are the majority population, but we don't want to miss that part.
Okay, we'll move to the next question, which is similar in a way from. Chang QH has some questions about large vesicles. Please unmute yourself and ask your question. Chang QH. If not, I'll read. I'll read the question. So, what about bigger size particles, and how big can you actually measure in the machine? So I know that you cannot put cells into the machine, right? That would block the machine, correct? But when is this?
We put a several micron size, let's say bacteria inside it, but I wouldn't dare to put the cell inside.
So what is the biggest size you can measure actually? So could you put in apoptotic bodies in there?
In case of EVs for the commercial one, I would say probably 200 to 300 size. Yeah. But if larger than 300, then that, no, it will saturate.
Okay. So Thierry, please unmute yourself.
But as I said, you can tune down the laser power or add an optical density filter. That can, you can shift the window to the larger size EVs.
Clotilde?
Yes, hi. Thank you for this great talk. So I think that one of the great possibilities of your machine is to do double label of single EVs because you have these two colors, right? I was surprised that you only showed one example of such analysis with the CD9, CD81 double stain, and where a majority.
We have that. Sorry.
Let Clotilde finish the question. And then we can answer. Go ahead.
Sorry, yeah. So in this plot that you showed, you had the majority of the vesicles that were double labeled. And I would expect that probably for CD9 and CD81 it's possible. I don't know, it depends on the cells, etc. But for other markers, I would expect more single vesicle EVs, single labeled EVs to exist. So did you do more extensive studies, and can you really see single labeled vesicles with the nano FCM? And if not, it would sound to me that there is possibly you know, some artifactual double labeling that can happen.
I don't know whether I catch your question. So most of the we are seeing single EVs. First, most of the time we do single labeling because we want to see if double labeling, I would say the statistical hindrance effect would affect. So we first do single labeling to see how much percentage can be labeled. And we also have data, I think in the 2018, the ACS Nano paper, I remember, so for, let's say, for CD marker, was a single labeling, it was originally, let's say, 58% positive. If you do double labeling, it decreased down to 38% because now it's very crowded. It's let's say EV are only probably 80 nanometer in size, and the antibody is 15 nanometer. And if you plus Vesicles restraint, so it's a very crowded environment that wouldn't allow efficient labeling of double labeling. If you want to do 3 labeling, that would be very difficult. Yeah, because compared normally people would say, okay, it's for the conventional flow, they have up to 15 or more parameters for single cell analysis, but the surface area of a 100 nanometer EV is about 10,000 smaller. So 10,000 copies smaller copy number compared to the cell, yeah.
So yeah, so you think it's not possible with the current tools, full antibodies, full-size antibodies, etc. It's not a good idea to use the nano FCM to do double label. Okay, thank you.
You cannot use other instruments because it's a problem of the material of the EVs you're working with. Yeah.
How many antibodies can you actually? How many different monoclonal antibodies can you use on this equipment? And this also relates to Aman's question: how many lasers and channels can we have?
Currently, we have a 2 laser system. Probably can do 3 color. Because besides as antibody labeling, you can also use the nucleic acid labeling or membrane labeling simultaneously with the antibody labeling. Yeah, so probably 3 fluorescence colors plus 1 scattering color, 1 scattering, yeah.
Okay, so Cheng Jie, I missed your question. Please unmute yourself and ask your question.
Okay. Okay, thanks, Professor. Can you hear me?
Yes.
Okay, thank you, Professor Yi, for your impressive talk. So I was wondering, because the current nano FCM the instrument is actually, to me, is more focusing on the accurate counting or numbering of different particles with different sizes. So I was wondering, are you going to maybe develop next generation instrument focusing on accurate sorting or even isolation? I mean, different particle of different size. I mean, it's like a column chromatography for us to isolate or purify the particle with different size. I mean, which is more challenging. I mean, yeah, I'm just wondering. Yeah.
Okay, yeah. Actually, this is a very good question. Every time I give presentation, people would definitely ask this question. I would say, okay, first of all, if you want to, of course, first, doing isolation, it's going to use a different technique. Okay, so make it more challenging. And the second part is, if you want to isolate the EVs for downstream biochemical analysis, I wouldn't recommend you do it, because think about the volume. It's 1 million smaller compared to a single cell. So even though at the same throughput rate of a sorting rate, let's say, of course, for us, we are a little bit slower than conventional flow. even with the same throughput, you would need 1 million more time to isolate enough quantity of material for downstream analysis. So what I would recommend is you use other methods to do isolation and use nano-FCM to do the quality control to help you to optimize your isolation technique. In that case, you can get plenty quantity of material for the downstream experiment, but you really need to use the nano-FCM to quickly give you feedback to guide you to which action or which strategy you need to take in order to get a more pure EV samples.
Okay.
I guess if you think about isolating 10 per second, it takes 10 seconds to isolate 100 vesicles, and
that's not enough.
and it just takes much. And when you need 10 to the 7 or 10 to the 8 to do something appropriate with the vesicles, so experiment functional.
It takes you a whole life.
It takes your whole life to go enough. Yeah, exactly. Is there any more questions here? I am struggling a little with equipment. I don't think we have any more questions. Carolina, do you have any more questions?
No, I think it was a very excellent talk, and I think Xiaomei explained very clearly on the advantage of using the nano-FCM, and also a lot of tips on which dye which we can use, and also tips for EV isolation. So that's actually plenty of tips for not just nano-FCM, but all those sort of information we can use for EV isolation in general. I think that was very good.
And we will put this on YouTube. But also we should put it on a Chinese channel somehow. Maybe you can help us with that, Xiaomei, so it can be accessible for the Chinese community as well.
If they want to go to YouTube, they should be able to do it.
Yeah.
Thank you very much, Xiaomei.
Thank you very much, everyone. And Carolina, it's your time to
for be the host. And if people have other questions, you can send me email offline.
Yes, definitely. Yeah, and
actually, Ekene Ankor is holding up his hand. You have a question, Ekene? Or maybe just applause, doesn't matter. Carolina, I think we end the meeting here. No more questions. Thank you very much, everyone. And Carolina, please sum up the meeting and introduce the upcoming events.
Yeah, thanks, Yan. So thanks so much, Xiaomei, for the presentation today. And also, thank you so much for having so many Chinese colleagues to come and join us. It was really enjoyable, and hopefully you can all join us next week as well.